This is actually the same question we had been doing the first two parts of it last class period. So this is that question we had ended with last time, and I went, oh, you should do this and have it ready for next time because I'm going to ask you about it. Okay. And so after the equivalence point, 30 milliliters, what's going to happen to your zinc? Well, almost all the zinc is going to be bound, isn't it? Okay, so you're going to have very, very, very small amounts of zinc remaining. And you're going to have to put in your dilution factor first. So if we figure out the complex concentration, then we can go backwards from the complex, and we can figure out the concentration of the free from the complex formation constant, can't we? Okay. And this is typically how you want to work with any complexes where you're going to be majority complexed. Most of it's going to be in the complex form. Okay. Because the complexation is going to be what determines your solution, right? How well it comes apart. Okay, and so we're going to take that dilution factor times the concentration we started with there, and then that's going to equal the 4.55 times 10 to the negative 4. Now we also know the concentration of our excess EDTA, so we again need to apply our dilution factor, which is 5 out of 55. And that's the concentration right there of our EDTA from the original problem. And that's that value. So the next thing we're going to do, well, once we know those concentrations, we can plug those concentrations in to our expression. And so we write up our expression. That's that complex right there, right? That's the EDTA right there, the excess, because that's going to be the major source of that EDTA. And then we are trying to figure out the zinc, aren't we? Okay. So we plug those numbers into here. We plug in our zinc. Notice right here is that mole fraction times the Kf, which is the Kf prime, isn't it? That we had practiced multiple times calculating. And so it's going to be 0.3 at this times 10 to the 16.5. Again, we get that from the table for zinc binding to EDTA. Okay. Where did the 0.3 come from? That's our fraction of the EDTA at pH 10, isn't it? Okay. Okay. And so we go ahead and solve. So we divide over, multiply, and we get zinc on its own. And that's going to give us 5.3 times 10 to the negative 16. We take the negative log of that, and what number do we actually get? Anybody want to re-answer? 3 of you want to re-answer. If you're not sure, feel free to re-answer. Okay. Again, if you take negative log of a number that's like 16, right? What do you get? You get 12. Okay. Got it. Okay. Is everybody done? There are now 3 people that are still wrong answers. I guess. Maddie, double check your answer sheet. Will double check your answer sheet. Olivia, I don't remember if you answered correctly or not. That's correct. Yeah, I mean, negative log of 16 something is 15.28. There's still one person that's answered wrong. I'm not sure who that is. Yeah, you both are right. Then you're right. Okay, now we're all correct. I don't know what to tell you guys at this point. Half the class gets it wrong, even if I give you the answer. Oh, guys. Oh, I am drinking the wrong thing in here. Um, all right. It's just going to be another day where I think I need to just lecture and. Oh, God. Should I start bringing coffee with me? Or? Oh. Okay. Anyways, 15.28. Um, so remember that after equivalence point, what you're looking at is you formed all of it into the complex, right? And so what you're really looking at is how much of it is going to fall back out of the complex. Okay? Okay. Now, let's talk a bit about the concentration of the complexing agent. So your greater concentration of the NH3, the smaller the observed change in your P zinc 2 plus near the VE. And so notice that the complexing agent, as we get larger and larger concentration, is going to go through this much faster, right? As we're lower concentration for the complexing agent, we're going to go through this much slower, okay? And so it'll give you a more distinct VE. Anybody know why you don't just use like 10 to the negative 10th concentration for everything? If you use such low concentrations, how much fluid would you have to have in that burette? More than the burette can hold, right? You'd have to have massive reservoirs, right? And huge solutions you make up, which isn't as much of a problem, but the having to refill the burette 20 times would be a. I'm sure you guys would be thrilled, right? Yeah. Now, so you do have to play a balancing act in all of these, that your auxiliary ligand has to be kept below a level that would absolutely obliterate your endpoint here, and that you can actually find a distinct endpoint. Okay. This right here is actually the titration of copper 2 plus with EDTA. We're using an auxiliary complexing agent. And so this is going to be your copper sulfate before titration. This one right here is the ammonia complex. Copper ammonias, by the way, have a just gorgeous blue color that's incredibly vibrant. So they're impossible to mistake with almost anything else. Okay, if you ever see the copper ammonia complexes. Now, this is buffer 10, pH 10 at the center. This is the endpoint right here. Once you've have the EDTA, where it's displaced the ammonia right here. Notice the color change. Changes from this very distinct deep blue color, right? And it's going to go to this much, much lighter blue, right? Okay, why is it going to a much, much lighter blue? Well, EDTA is displacing the ammonia, right? The ammonia copper is what creates that blue color. Okay. Okay, so let's talk about metal ion indicators. This is actually the most common way to detect the endpoint in EDTA titrations. The compounds change color when they bind to a metal ion. Must bind metal less strongly than EDTA so that they can be displaced by the EDTA. They're also going to be acid-base indicators, so you have to control the pH. If you allow your pH to change, most of these indicators will change with that. Okay? Because typically they're going to be like negative ions that could take on NH+, right? For the most part. So if they get protonated, they're going to change. The metal ion indicators can be used only in certain pH ranges, just like you would have talked about the different pH ranges that you can use for the acid-base indicators. Now, some of the endpoint detection methods are going to be metal ion indicators, of course, which we've been talking about right here. We can also use ion selective electrodes. Did you remember that copper 2 plus in the superconductor? It said we could have used a selective electrode for copper, right? And that would have worked as well. In fact, next time around, next time I teach this, I'll probably have I'm gonna probably shuffle a lab in where we actually make an electrode, or maybe next semester we'll do that. We'll actually make a copper selecting electrode. Okay. You can also use a glass pH electrode as well. It works similarly. So this right here is going to be an example of this. We have magnesium. And so this is going to be a titration magnesium. This one's B4 right here. In this case, we're going to use Aerochrome Black T as our indicator. And so here at the near the endpoint, we're going to be this purple color. See how we're going from red to blue? And so we're purple. And this is telling us, hey, we're near the endpoint. And so you would slow that down, right? And start adding your drops. Okay, and watching the color go and disappear, right? As you mix it. And so after the equivalence point right here is the right. Now, you can actually use Calgamite as well. It gives very similar coloring to this, and it gives a very similar effect. Okay. There's actually a variety of reasons you would want to use different colorings, by the way. But this right here is what's happening. We have magnesium in the indicator, right? It's a red color. Okay. EDTA is colorless. Magnesium EDTA is colorless. It's our indicator that goes back to being blue when it's no longer bound to the metal. Okay. So when we titrate away our metal, because EDTA binds to it better, we're going to end up reducing that color that is the blue red color, excuse me, and we'll end up in the blue color. Okay, this is actually what those concentrations would look like. This would be magnesium calgamate right here, and see its wavelength is this 550-ish. And then we look up here, and this is the free calgamite right here. And so that would have a wavelength closer to about 600 something, right? 630-ish maybe. Okay. And so, yes, you can monitor it by eye, but your eyes are not super sensitive to color changes. You can actually monitor it using UV vis spectrometer. In this case, you could get away with just a visible light spectrometer. You don't actually need a UV, okay? Which is a cheaper thing. Has anybody used the visible light spectrometers in the labs at all? Okay, we'll use them next semester. You didn't use them at all in general chemistry to do Beer's Law or anything? No? Okay. Okay, wow. That's okay. We definitely need to get one in next semester then. Oh, wow. All right. So let's look at color plate 7. So too much indicator makes it hard to discern color changes. Subtle colors like the red 1-to-1 bismuth, pyrocatechol violet complex can actually signal that the equivalence point is close. This is why it always recommends you in any instructions to do like 2 or 3 drops and not do, tons. Just put in a milliliter, right? Because you lose all that subtle coloring that you would see because the concentrations of the indicator end up so high that you either have the indicator or you have everything's bound. Okay? Whereas you can see actually different colorings depending on your different concentrations. So this right here is actually titration of Bi3+ in Pepto-Bismol caplet powdered by EDTA. This is using the pyrocatechol violet indicator, and this would be at pH 1.9. And you can see here we get a very nice pure purple starting color from 2-1 as we go along. And then we actually can get the intermediate shades of the violet here. And you can see these set of intermediate violet shades here. And then it starts going, approaching the endpoint where you have this right here, which is the appearance of the 1-to-1 BI indicator. And then you actually end up at the pure yellow endpoint color. And so this right here actually warns you, hey, I need to start going drop stone. Okay. And so if you put in too much concentration of the indicator, you would not see all this. You would only see this, and then all of a sudden, bam. Okay? It's unlikely you'd see anything between. So this is indicators that can be used in only certain pH ranges. Xylitol orange changes from yellow to red when it binds metal ions under acidic conditions. This is actually a really easy one to see because it follows our eyes color palette really, really nicely. Unless, of course, you're red blind, in which case. No, it is not quite as easy to see. Can everybody see red, orange, yellow? Okay, cool. All right. Right here, we see some common metal ion indicators, very, very common ones that you would see in a lot of these titrations. These right here are their pKa's. That tells you what form you'd have available at these different pHs, right? And then this right here is going to be the colors of those. And so, anybody have any questions on how to read that? Okay. Right, now, remember that when you do this direct titration, you're stripping the metal ion from the indicator, right? And so the indicator has to give up its metal ion to EDTA. If the metal does not freely dissociate from the indicator, the metal cell will be said to be blocked. So it'll be blocked to the indicator. Now, iridium black T is actually blocked by copper 2+, nickel 2+, cobalt 2+, chromium 3+, iron 2+, and aluminum 3+. If you were to use it with this indicator with any of these, it won't actually come off properly. Okay? And so it can only be used for back titrations of these metals, not direct titrations. If you try to directly titrate it, the metal ion will not come off, and you may get some results. It may be difficult to actually realize that you did not get any results, but you will definitely get wrong results. Okay? That is not to say none of the metal will come off. That is to say a significant amount will not come off. Okay? Okay. So, direct titration. This can be where the analyte is titrated with standard EDTA. Analyte has to be buffered to ensure large conditional formation constant and distinct color changes. You do not want your pH to change because it'll change your indicator, but it'll also change the concentration of EDTA, right? So you need to make sure that you're always doing these in buffers. And they need to be probably like pH 10-ish buffers typically. Okay? Though you can do it pH 8 and such. It's just you have a lot less EDTA available. Now, metal complexation by EDTA must be rapid and go to completion. Would you want to do something that complexation takes for like a couple minutes each time you put stuff in? Does that sound like a fun titration? Drop. Yeah, I'll be back in 5 minutes. Drop. 5 minutes later. No, that sounds like you're never getting done, right? Okay, so rapid titration, rapid complexation also needs to go to essentially completion. Now remember, they'll never actually go to completion, but they'll go to, 10 million times a million times a million completion, right? They'll go to 10 to the negative 14 sort of completions. Now, auxiliary complexing agents may be used to prevent metal ion precipitation. Keep that in mind, that if you have something that precipitate out in water because of the hydroxide, you can put something like ammonia in there. You can put an ethyl ammonia, things like those, to complex it instead. And that will keep it complexed until the EDTA takes it away. Phytoremediation. So this is actually your approach to removing toxic metals from contaminated soil. One of the ways to do this is actually to grow plants that accumulate 1 to 15 grams of metal per kilogram of dry mass of plant. Does anybody know what some plants that are really good at this are? Nobody's heard of this before? Sunflowers are a really good one. Lettuce is also quite good at it. Did you guys know that they oftentimes will sell human fecal matter as fertilizer materials? Okay, do you know why they don't do that very often, though? Contamination. The toilets also tend to have a lot of metal ions that end up in heavy metals. And so food grown with it tends to uptake the metal ions, and they poison the people that eat it. Okay? And so they've contaminated large quantities of land, actually, with metal ions that they're having to remediate now. Not all because of that. Some of them just because of spills or lots of melting activity in the area, that sort of stuff. Now, so this plant is harvested to recover the metals. It can be things like lead, cadmium, it could be chromium, it could be whatever, heavy metals. The metal uptake can actually be enhanced by adding EDTA to mobilized insoluble metals. EDTA is used, though. Care has to be used to prevent the soluble EDTA metal complexes from leaching into the groundwater. If you allow it to leach in the groundwater, it will be soluble for a while. It will eventually have the EDTA destroyed, and it will come out in places you do not expect. Okay? So keep that in mind that if you use EDTA, it will mobilize these insoluble metals. But if you allow it to drain out to areas that you don't want to contaminate, you will have contaminated those areas. Okay? Being mobile is dangerous. Okay. Now, back titrations. Anybody done a back titration? Okay, let's define it quickly, and then you can see. You know the excess of EDTA that's added to an analyte. The excess EDTA is then titrated with a standard solution of a second metal. This can be necessary for several reasons. Your analyte precipitates in the absence of EDTA, and so you can get away without using a secondary complexing agent by just complexing everything with EDTA and then seeing how much excess EDTA you have. Okay? The analyte reacts too slowly with EDTA, and so if it reacts very slowly, you can set it up, do the reaction with all the EDTA you're going to use, and then use a metal that will quickly bind to the EDTA to back titrate it. Okay. The analyte blocks the indicator. So if your analyte blocks the indicator, of course, you can go ahead and use this with back titrating it with the EDTA. Once you get past the amount of EDTA, then it'll start binding with the indicator. Now, the metal ion for the back titration must not displace the analyte from the EDTA. There needs to be a significant difference in their binding between what you're looking at and the metal you're going to use to do the back titration. Does everybody know what a back titration is versus a forward titration? Or normal titration? Okay, so a back titration versus a normal titration. Let's say you have this much of your analyte, right? A forward titration, you're just going to put in EDTA until you detect the endpoint, right? And so you would then titrate up to that endpoint, right? And however much EDTA you put in, that's the amount of analyte, right? Okay. A back titration, you're going to use a known quantity of EDTA, okay? And then you're going to mix it with your analyte. It's important that your amount of EDTA is in excess of your analyte, okay? And then what you do is you take another metal ion of some sort, something that won't bind better than your analyte, and you go ahead and titrate until you reach your endpoint. And so this is then going to be that titrant, and this would be the other metal. Okay? And so what you would do then is you're back titrating. See that? You're actually going backwards. You're going how much EDTA is left after I put it in. Okay? Now, having said that, back titrations are actually pretty commonly used alongside regular titrations. So let's say I did my titration. Whoops. Overdid it. And, super pink with my phenolphthalein indicator, right? I over titrated. I messed up. My finger slipped and I deposited like a couple milliliters of my titrant in there, and oh my goodness, what do I do? Okay. So what you can do is you can actually do a back titration. So this one was forward. This then would be the back titration. So let's say you had titrated with NaOH. You can take a burette with a known concentration, a standard HCl. You can add that until you go back to just past your endpoint. You subtract these off then, and that will give you your amount of that you should have titrated, right? So very, very common methodology to use if you need extremely precise results, but your endpoint's difficult to actually determine. Yeah. If you have an indicator that you're not very practiced with, if you have an indicator you can't see very well, reasons like those. If your reaction tends to be a little slow, it's also a really easy way to do it. Okay, so this is actually a back titration right here. Nickel 2 plus can be analyzed with a back titration using the standard zinc 2 plus at pH 5.5 with xylenol orange indicator. The solution containing 25 milliliters of nickel 2 plus and dilute HCl is actually treated with 25 milliliters of the sodium 2 EDTA. The solution is then neutralized with NaOH, and the pH is adjusted to 5.5 with acetate buffer. So the solution will turn yellow when a few drops of the indicator are added. The titration with 0.02299 molarity zinc 2 plus would require 17.61 milliliters to reach the endpoint. What would be the concentration of the nickel 2 plus? So notice you add a standard amount of EDTA. See that? Okay. So then you titrate it away, the EDTA that remains, with zinc 2 plus. So whatever the difference between the moles of this EDTA here and the moles of zinc 2 plus, that has to be your other metal that binds to EDTA, right? Okay. So we go ahead and do the math. Again, how do we get the millimoles of EDTA? Well, we take the milliliters times the concentration, right? That gives us millimoles of EDTA. We get the millimoles of zinc. We take the difference between those. And we're going to get the millimoles of nickel. That's all a back titration is. Did I lose anybody there? Okay. Once you have millimoles, you can always find out concentration by just taking millimoles divided by milliliters, right? And I'll give you a concentration of the nickel 2 plus in this case. Okay. Notice that back titrations are not necessarily that much more difficult than a forward titration. There's a lot of advantages to them, in fact. Now, let's talk about displacement titration. The metal does not have satisfactory indicator, but can be displaced by a metal that does. And so mercury 2 plus has no indicator. It can be displaced by magnesium 2 plus bound to the EDTA. Our newly free magnesium 2 plus has several metal indicators that can be titrated with EDTA. In fact, I've used 2 of the indicators as examples in this lecture so far. It shows you how common indicators for magnesium are. And so this is actually the reaction we would see here. If we have mercury 2 plus, it's going to displace the magnesium 2 plus, freeing it, and then that will then bind with your indicator, right? And so we can do a forward titration. It's just a displacement titration in this case. Okay. Silver has no indicator. It will displace nickel 2 plus bound to tetracyanonickelate. The newly free nickel 2 plus has several metal ion indicators, of course, and these can be titrated with EDTA. And so right here, we actually are doing a displacement off of this tetracyano. And so the silver is displacing the nickel. once the silver has displaced all the nickel, we'll have our maximum concentration of the nickel there. All right, so let's look at indirect titration. So this is going to have anions that precipitate with certain metal ions. These can still be analyzed with EDTA. So we have sulfate. This is precipitated with excess barium 2+ at pH 1. The barium sulfate is then going to be washed and redissolved in excess EDTA at pH 10. This will obtain a soluble barium EDTA complex. And so the excess EDTA is then going to be back titrated with magnesium 2+. And so we're going to actually do an indirect titration of the anions using cations that bind them and precipitate them. Okay, so we can isolate them, right? So this is how you would do EDTA titrations to detect anions, which brings in the entire almost world of organic chemistry, right? Because you can typically generate an anion out of most organic compounds. Okay? So this is kind of a cool way you can actually work in a whole nother set of chemistry by using these indirect titrations with EDTA. Like I was mentioning, EDTA, very, very powerful technique, EDTA titrations. Okay? They take a little bit of while though. So instrumental techniques have mostly replaced most of these methods. But when you need to verify something, or if you have one-offs things, or if you have tests that are just not working on your instrumental methods, this is a great tool to have in your pocket. Now, masking. Masking is trying to prevent one species from interfering in the analysis of another species. Note that masking is not an EDTA-specific thing, or even a titration-specific thing. It is a analytical methodology-specific thing. Okay? It's in fact, it's not even. It also shows up in organic chemistry. Okay, you may remember masking if you took my organic chemistry class, where we took functional groups and we masked them so they didn't react. Okay, masking agents going to protect some component of the analyte from reaction with EDTA in this case, though it doesn't have to be an EDTA titration. Aluminum 3+ can be masked by fluoride. Cyanide masks all of these metal ions, but not these ones. I'm not even going to bother going through that. Demasking. So what demasking is taking the mask off, okay? And so it's going to release your metal ion from the masking agent. Cyanide complex can be demasked with formaldehyde. So you put the cyanide in with them. They'll mask them. Then you can do your titration of one of these ions right here. And then after you're done with it, you can go ahead and demask it using formaldehyde. And that'll give you back your metal ion. And then you could do the titration of that one. Okay? And so you can somewhat selectively do detections of these. Okay? Now keep in mind, this is actually a major deal, right? Because if you titrated the mixtures of these things, you would get all of those titrations together, wouldn't you? Okay? And you wouldn't really be able to tell which one's which. Now, water hardness. This is the total concentration of alkaline earth ions in water. Predominantly, it's going to be calcium 2+, magnesium 2+. The hard water actually reacts with soap to form an insoluble product. That's that precipitate right here. People typically call it soap scum. This is typically if you wash your dishes and it doesn't work very well. This is probably why. You probably have hard water. Okay. Note that it doesn't technically need to. Hard water actually in hardness refers to the alkali earth metals, but there's also other metal ions that can cause the same issues. Okay. Now, hard water leaves these deposits. They're called scale-on pipes when it evaporates. Iron, zinc, copper, cobalt, and other metal ions will also interfere with the hardness determinations. Because they'll bind to the EDTA, they'll obscure the endpoint color change. So in standard methodologies, you actually add cyanide or sulfide. This will minimize your interferences. They'll mask these other metals, and you'll just be left with your calcium 2+, magnesium 2+, that you can then do an EDTA titration of. Okay? And then you can selectively do titrations and detections of only one of those later. and get a difference. So this is actually biomolecular binding. You'll notice that biochemistry has heavily now used these same techniques. Whether most people now use them actually understand where they came from is questionable, but they use these exact same techniques now heavily. So biomolecular binding is highly specific, and that's because your typical ligand binds very specifically to that enzyme, right? This is the lock and key model, induced fit model. Does anybody not know these models yet? So lock and key model, the substrate fits in like a key into the enzyme, and the enzyme doesn't change. Okay. Induced fit model is where the ligand, the substrate, binds to the enzyme, and the enzyme actually has to change to fit it better. Most enzymes work much more like an induced fit model, though there's a bit more to it now because the overall shape of the enzyme oftentimes changes as well. Okay, but I think that's enough for this class. If you're really interested, we can talk in like biochemistry and stuff. Check out biochemistry. Now, affinity constant, this can be called the association or binding constant. Notice it's the same sort of binding constant we've been talking about this entire time. It's going to be an equilibrium constant for your reaction for your biochemical receptor with a target ligand. It's going to be this value right here. Typically, again, be measured under physiological conditions, which is why the prime is there, is because it's a modified one under physiological conditions. Okay, and so this would be that right there, the complex divided by the free ligand and the free enzyme concentration. Okay, so what are some of the classes of analytically useful biomolecular binding interactions? Well, you have antibodies. You know, how well antibodies bind is going to affect how well your body actually responds to it, or if your body responds at all. The aptamers, this DNA or RNA, so epitome on small molecules to proteins. We have biotin as your ligand right here, binding to this protein. We have enzymes, of course, binding to different cofactors, substrates, inhibitors. A lot of vitamins are cofactors, by the way, in case you're curious what is going on there. And I think you guys have enough here. Do you guys care anymore about that? If you do. Anyways, those are all binding constants, right? So this is actually an aptamer. It's going to be a short length, which is 15 to 40 of the single or double-stranded DNA or RNA. It'll strongly and selectively bind a selected molecule or feature of a protein. An aptamer for a desired molecule is going to be chosen from a library of about 10 to the 15th random DNA or RNA sequences by successful cycles of binding to the target by removing on-bound material and replicating the bound nucleic acid. This is going to be a process that's called SELEX, systematic evolution of ligands by exponential enrichment. And so right here we actually have a fluorescent dye. This is bound to this structure here. This right here, this stem, is going to be the closed part right here. That is going to be close to the binding pocket. This right here is the binding pocket right here. And so when you have these, what's going to happen is they're going to open the binding pocket. They're going to bind to whatever you're looking at, say fentanyl. Okay. Once they bind to fentanyl, they're going to label it with a fluorescence signal that you can then activate. And so you can shine a light on this after applying it, and you would see fluorescence. And hey, that's fentanyl. Okay. This is a very typical way to, you can actually put these on binding beads too and flow things over them. So this is actually a very, very useful array of tests that you can do. There's a lot of these being developed, in case you're wondering. So ligand binding assay, this is going to be a receptor ligand binding assay, an analytical procedure which is going to rely on the equilibrium binding of ligand molecules to receptors like proteins, antibodies, aptamers. The reagent form of the fentanyl aptamer will consist of multiple components. That's fentanyl right there. And so again, this is going to be your strand of complementary DNA right here, which the aptamer is open. You'll have a quenched Q right here The quenched Q right here is important because this is your fluorescent agent. If it binds properly, what will happen is your fluorescent agent will be activated, and that will create a fluorescent compound. If it's always fluorescent, by the way, you will have troubles, because then you will always have a fluorescent compound that's in everything you do. So it'll give you a lot of false positives. Okay, this right here is going to be the calibration curve for the ligand binding assay of fentanyl right here. And so the analytical curve follows the general form of a 4-parameter logistic function. And so your ligand binding assay response is this equation right here. Yi is actually the signal. That one right there. A is going to be the limiting signal at low concentration. And then you're going to have D, which is the limiting signal at high concentration. That's where it would max out, right? You'd end up with a 1. And so Xi is the analyte concentration down here. And then C is going to be the concentration, the inflection point of the curve right here. This is where you're going to have this reach this inflection point here. And then B is going to control the transition between the two limiting limits. This is typically called the growth factor. If this is a larger number, this is going to end up being much steeper. Okay, so let's talk about immunoassays. This says antibody is a protein produced by the immune system in response to foreign molecules and antigen. Immunoassays, or IA, is going to be an analytical measurement based on the binding of a specific analyte by an antibody. You can typically actually set these up by doing things like injecting small quantities into mice over time, and they'll start developing immune responses to it. And then you can collect their antibodies, which will then bind to that substance. You can also there's some pretty cool like genetic studies where they're designing these things. This field has actually had a tremendous amount of growth in like the last 10 years. So if you're ever interested, it's actually fascinating to look into. They've done some pretty crazy stuff, like make drug-specific ones that you can actually take as medicines and won't let you get high. Which is pretty crazy stuff. So the affinity constant for an antibody-antigen complex is very large. The Ka prime of the antibody to other molecules is very small. And so what you would see is that these very selectively bind to the target. Okay? Whereas they bind very little to anything else. They're very, very specific. This is what an antibody looks like, by the way. It's composed of 4 parts. You have the heavy chains and light chains. Okay? These are binding sites right here. Anybody know what these are? They're called the disulfide bridges. They're 2 sulfur groups covalently bond together. That's how they hold together. And they'll get cleaved later. Okay. This is enzyme-linked immunosorbent assay, or ELISA. This right here is really, really popular in biochemistry right now. Do you guys do any of this in biochemistry? Maybe you'll do it next semester. I'm not sure if you guys do or not. Anyways, it's a biochemical assay and incorporates an enzyme attached to an antibody. After the antibody binds the analyte, the excess antibody is washed away. The reagent is then added to convert it to a detectable product by the enzyme. The quantity of detectable product is then proportional to the amount of analyte. And so here you have this polymer, like usually it's a column and it has little polymer beads in it, more often than not. And it has antibodies that are deposited on those beads. Okay. And so then what happens, you add the sample containing your analyte. It's going to then be washed and remove the unbound molecules. And so here you have your analyte bound to the protein, bound to your antibodies here. Okay, so you've attached them onto your beads, right? Washed away the excess. Okay. And so the next thing you're going to do is you're going to add an enzyme labeled antibody 2. you're going to wash to remove on-bound antibody. And so right here is your enzyme covalently bound to the antibody 2. Okay. So the next part right here is that this is going to be undetectable at this point. However, when you put something through that's an undetectable reactant, remember this is an enzyme, right? It's going to act as an enzyme on that reactant. And so then you can convert that to detectable product. And how much you convert to detectable product is proportional to, well, the enzyme bound, right? The enzyme bound is proportional to, yep, the analyte protein, isn't it? And so, I know, right? It's a little indirect, but keep in mind, these proteins are huge, right? They can be very, very difficult to detect, and in many conditions that you could detect them, they're going to be very difficult to actually test. Okay? And so what this does is you have your resin beads with the antibody. They're going to bind your analyte protein, and then you have another antibody that binds that analyte protein on the other side. That has enzyme on it. That enzyme is going to allow you to make something you can detect. Typically, you can wash these off. You have a solution you can pass through to cause these to unbind from the antibody to some degree. But even if you lose them, I guess you've only lost so much. Okay, so let's talk about how a home pregnancy test works. So this is going to be your urine containing HCG. This is your sample pad right here that you apply it to. And it has antibodies bound to silver particles right here. This is going to be the antibody to analyte right here, which is the test line. And this is going to be your control line, which is the antibody to antibody right here. This portion is a nitrocellulose membrane. This is another absorbent pad right here. And this is the conjugate pad is what this is called. Okay, so first thing you do, you apply the drop of the urine to the sample pad here. That HCG is going to bind to your antibody as your liquid wicks past the conjugate pad right here. Okay. And then this part of the HCG is going to bind to the antibody at the test line. Once it starts binding to the antibody at the test line, the conjugate reagent will not be attached to the HGC, and it's going to bind to the antibody at the control line. And so what you're going to see here is you start binding to this line here, test line, and then it'll further release that conjugate, which will then start binding to the control line. Okay. And so right here's the sample pad, up here's the test line, and there's the control line. You get your double line, and then you go either yay or oh shit, depending on what your plans were. Hopefully yay. Okay. Hopefully. Competitive immunoassays. So these are going to be immunoassays which the analyte competes with a constant amount of labeled analyte for a limited amount of antibody. This can be used when the analyte is too small to bind to two antibodies. So you can't do the sandwich assays up here because it's just too small. You won't be able to fit that number of antibodies around it. It's going to test for things like drugs, toxins, antibiotics, pesticides, all of those things. Now, what we see here is we have an antibody, again, put down on some sort of surface. This could be a resin, this could be a plastic film, it could be kind of anything you want, really. Usually put in some sort of package, you can kind of dip the end and wick up stuff into it. Or done as a more formal like column technique. Okay. Right here is going to be the ochratoxin A labeled with your enzyme. And right here is ochratoxin A right there without the enzyme. And so what you're going to see is you're going to incubate this. And that's going to start attaching the ochratoxin A to the antibody. Then you're going to rinse off any excess. And then you're going to add your enzyme substrate. And so the detectable product is then going to be converted using your enzyme right there. You're then going to take and allowed an enzyme substrate reaction to occur for a fixed time. Why is it important to do a fixed time, by the way? An enzyme is a catalyst. So, like, if you just let it do 5 minutes versus 10 minutes, well, it could convert twice as much, right? So it doesn't matter exactly how long you let it do it. What is important is that you do it for a fixed time that you know the results of every time. Okay? That's the important part. Now, this first part right here, you have a known amount of the enzyme labeled oxytocin. And so as you add more of the ochratoxin A from your sample, right, the stuff that's labeled with enzymes has less and less ability to lay down, doesn't it? There's less and less chance that it binds. So this one's actually going to have a reverse concentration gradient, right? As you have higher concentration in your analyte, you're actually going to have less of the enzyme-bound stuff binding. Okay? Because it's going to have to competitively bind the antibody. Does anybody have any questions about that so far? Okay, so what are some of the analytical characteristics of the immunoassays? Well, the main reason that they're done is the low cost. Okay, overall, they typically are lower material costs than most of these things. You can usually bulk them up to very large scale production, and their costs go down tremendously. But the biggest cost savings for you is typically going, at least a business or a person, is going to be the lack of you guys. I mean, think about it, right? You're going to want a decent salary, right? And so if each of these takes you 10 minutes, then it's going to be pretty expensive, isn't it? Okay? If each of these takes, literally, you take 10 minutes to verify a batch of 1,000 or 1,000,000 of these, then it's a lot cheaper, isn't it? Okay? So they generate a lot less waste than most laboratory techniques, especially like liquid chromatography generates tremendous amounts of waste. They're compatible with remote and field analysis, so you can take these out to places, you can store them under suboptimal conditions, and you don't have to buy a $50,000 instrument that could go with you. And so much more portable. They can perform multiple analysis simultaneously. You can have lots of strips that you give lots of people, and they can do it all over the country or all over a city. Their precision's typically about 5 to 25%. Now, that is not a very good analytical precision, is it? But in like a clinical decision or screening samples in the field, these are pretty good. Like, especially once you start doing it a few times, right? You can get good enough. They have good to excellent selectivity depending on your antibody. These things can have amazing selectivity. In fact, many times you'll see these analytical immunoassays done alongside traditional techniques just for their selectivity, because they can just be amazing selectivity, do a great job doing things like masking, all sorts of the other parts of the techniques I've been talking about, right? Okay. So really cool techniques, a lot of research being done on them, a lot of development lately, okay? I'm going to be honest, even if I made a lecture talking about all the most recent things, by the time we actually got to the lecture, I'd be like a few thousand publications out of date within a month or two. This is a huge field right now.